So welcome to the Woodpeckers morning concurrent session. I think this is kind of broadly a lot of wildlife talks, and so it's great to see so many folks here. I am Sky Greenler. I'm a PhD student in the firm department studying wildfire, and I'm going to be moderating this session and getting to kind of introduce all of these great speakers and then facilitate questions and answers, which will be just a little trickier on Zoom, but we're all quite used to it at this point. So if you have questions for the presenters, as I said, you can either type them in the chat and I'll ask them to presenters, or you're welcome to raise hand and I will unmute you. And then finally, if you're willing to give feedback to the presenters, we would really appreciate this. Because this is a student symposium, we really push giving constructive and positive feedback to presenters so they can kind of learn and grow into the future doing presentations. So each presenter has their own feedback link that you can access on our conference main page to do that. And if you are a student, for every feedback form you fill out, you get entered in a raffle for our $50 Peak Sports gift card. So that's a pretty good prize. And there's 6 of those, so the odds are good. But I want to start by introducing our first speaker, Mark Kerstens, who is going to be talking about black-backed woodpeckers moving between the green and the black, natal dispersal and survival of juvenile black-backed woodpeckers. And Mark is a student in the firm department. So Mark, take it away. Thank you for starting us off.
Sure, thanks for that nice introduction, Sky. Hello, everyone. Thank you for tuning in for my talk here. So I'm going to be talking about, well, my title that Sky read off for you, and then also more broadly, just black-backed woodpecker breeding demography. And so here we've got a nice side-by-side of 2 very contrasting images. And clearly you can see there are a lot of differences in the structure of these 2 forests. We've got the burnt forest and the green forest. And what if I told you that our species of interest, which is thought to be a highly specialized burn obligate, actually uses both of these areas in our study area? So some questions to consider as we move forward. What is a black-backed woodpecker? And how are they impacted by breeding in green versus burned forests? Specifically, their apparent nest success, their juvenile body condition, and their juvenile survival. And how do their used nest sites in green forests compare to their used nest sites in burned forests? And then how do juveniles disperse and choose their breeding habitat on the landscape scale? So the black-backed woodpecker is a non-migratory resident bird, and specifically the ones we're looking at are located in the south central Oregon in the Fremont-Waineba National Forest. They are a keystone species and an important indicator of forest health. Nest cavities are used by more than 65 vertebrate species in the Pacific Northwest. So you can kind of understand why it is so important to have these guys in your ecosystem. And so the black-backed woodpecker, they are cavity nesting birds, and they are primary excavators, meaning that they excavate the cavities all on their own. And a big part of their diet relies on these nice juicy bark beetle larvae that they use to feed themselves and their young. And so they're often associated with beetle kill forests and wildfire because of the high amount of standing dead wood or standing and decaying wood that they can easily excavate. And so, as I mentioned, our birds have been found to use the green forest, albeit in lower densities than in the burnt forest, but they are still breeding there. So for our data collection, we went out and locate and monitor nests to quantify their nest success in the 2 forest types, burned and green, and we record video of adult provisioning behaviors at the nests. And we measured and radio tagged nestlings right before they fledged to quantify their survival. And then we also measured the vegetation at nest sites to assess their habitat use and selection. So how do fledgling black-backed woodpeckers differ between the green and the burned forests? So apparent nest success in both of these forests is quite high, with 83% in the burned forest and 86% in the green forest, even though one might expect going into this that the birds in the green forest would have lower nest success because there's going to be, or at least we would predict that there would be a lot less of the specific type of food resources that they would want to eat and feed their young. And so if we take a look at body condition, and so if we take their body mass and regress it on a size metric, here we used build length, it can kind of tell us as a proxy how much fat reserves that the birds have, taking into account their size. Now, this specific measure that we've used here is a little bit biased towards larger individuals, and so I'm just using it to show some preliminary suggestive results, and we'll do some additional analyses to further tease this apart. But at least for now, we can kind of look at this and see that birds in the green forest might have a better body condition than birds in the burned forest, which is certainly interesting. And now, we may want to take a look at the data behind that a little bit. And it's important to note that these individual birds are not necessarily independent variables because we'll have multiple birds in the same nest. And so if we look at the average body condition by nest, the nests in the green forest are still a little bit better than the birds in the burned forest, but it's quite marginal, and they're basically the same, which again is still a very interesting result. And then the average number of birds fledged is higher in the green forest, but the standard errors are quite broad. And so again, they're quite similar. I guess it would be the takeaway from that, at about between 1.5 to 2 birds per nest being fledged. And if you look at the average max clutch size, so that's the number of eggs that you had maximum, the burned forest has a tiny bit higher than the green, but again, quite comparable, especially when you can take into account the standard errors. And it's important to note that the sample size here is a little bit smaller because than the average number of birds fledged, because for the average max clutch size, there were several nests that had unknown max clutch sizes. So if we look at their apparent survival, again, very similar between the two, about 60% average apparent juvenile survival. Now, you may be interested in how long did they survive? Is there maybe a difference in did one of them survive for less amount of time, over the time that we were monitoring these birds after they left the nest? And actually, yet again, the pattern holds similar between both the burn and the green forest, where the green circles are nests or juveniles, that had no evidence of a mortality. The yellow circles are where we had a confirmed raptor mortality, and the red circles are a confirmed mortality, but we are not sure what caused it. And so, and the exposure days is just how many days that they were available until the last time that we were able to detect them. Now, another part of this puzzle that'll be interesting and maybe help tease some of this out is measuring nestling provisioning. And so that's how much food and how often that the parents are bringing this food to the young. And so to do this, we've got these nest cameras, and I've got a few interns who are moving through this footage to answer some of these questions. Are there differences in overall provisioning rates between green and burned forests? How do males and females provision differently between green and burned forests? So this will be an interesting piece of the puzzle to add in once that data is finished being collected. Now, moving on to habitat and nest site selection, how do these compare between the green and burned forests? So to quantify this, we measured the vegetative indices and the nest tree in a 10-meter radius around the tree, then picked a random azimuth, went 25 meters, picked the nearest tree that would be suitable, and then repeated the measurement, then went 180 degrees from the random azimuth and 25 meters from the nest tree, and repeated the measure again. So we've got 2 available trees for every used tree. And I used some modeling procedures in order to determine what were some of the most important predictor variables. And one of them was the decay class of the trees. So decay class was most important for the green forest birds, and it was predicted that stage 2 declining trees that are still alive would be the most selected and most indicative of being used. And if you look at the data above that, you can kind of see most of the birds in the green forest used stage 1, stage 2, and a little bit of stage 3 and 4, whereas most of the trees in the green forest, at least the ones we measured, were stage 1 of available. However, if we now take a look at the birds in the burn forest, we can see that this relationship doesn't really hold true as much anymore, and it seems like the birds basically selected trees that just kind of mirrored what was available to them. And the modeling kind of emphasized this as well and said that there was no relationship for decay class in the burned area. Now, if we look at number of cavities in the used versus available trees, we can also see, and so it's important to note here that this does include the new cavity that the birds excavated that year. But so if we look at the tail end on the right side of this figure, we see some of these birds are selecting trees that already have a few cavities. And so having cavities in the tree already, it may be indicative to these birds that this tree will be good for excavating and a good nest tree. And so another year of data collection can maybe help to tease out this relationship a little bit more. Whereas, you can see that in both the burn and green forest, the available trees in most cases did not have any cavities at all. So we've got some questions about natal dispersal, and that's where are they moving to? So this is after the juveniles leave the nest and over the course of the year to where they are breeding. Where do they disperse? How far? And are they breeding? And so this is both for birds from the burnt forest and the green forest. And are they staying in their same forest type as well? And so we've got some pilot data from 2019 to 2020, where we just tagged a few birds with these delayed activation telemetry tags, where we tagged them in 2019. In 2020, we relocated them. And so in the burned forest, we had one bird that just moved 350 meters, and then we found it breeding with chicks the next year. In contrast, in the green forest, we had two birds that were tagged in 2019 and moved a respective 19 and 6 kilometers, and that's actually the satellite imagery of where they moved. And neither of them were breeding. And so obviously this is a tiny sample size, and so it'll be good to tease this out a little bit more. And this year, instead of using regular telemetry tags on our nestlings and tracking them just in the summer, all of our nestlings are going to be getting these CTX tags. And we have adjusted the activation schedule so that we can tag them in June and then track them actually through the summer, which will give us comparable data to what we've had from 2018 and 19. And then these tags will turn off. And in October, they will turn on for 2 weeks, and we can see where they have moved in those 2 months. And then they will turn off again and come back on in June of 2022. And then we can relocate them to further tease this out and really get at some of these dispersal questions and really tie all the different pieces of this puzzle together. So some preliminary conclusions. How are these birds impacted by breeding in green versus burnt forests? Their apparent nest success seems to be very similar between the two. Their juvenile body condition is potentially improved in the green forest, but still pretty similar, and hopefully more data will help us tease this out a little bit more. Juvenile survival is, again, also very similar between the two. And parental provisioning of the nestlings will be to be determined. How does their use of green forests compare to burnt forests? They use different cues to select optimal nest sites for a given forest, and that probably seems kind of obvious given the glaring structural differences between these two forest types. But maybe not for a comparison standpoint, but it will be interesting and important to understand within each forest type what cues these birds into a good nest site selection, because that will be important information for managers, both in green forest and burned forests. And finally, how do juveniles disperse and choose breeding habitat on the landscape? Again, this will be to be determined, so stay tuned. I would like to acknowledge everyone who has helped out on this project, both with logistical and funding support. And I will open the floor for any questions. Thank you very much.
Thank you, Mark, for that great presentation. So folks can put questions in the chat or raise your hand and I'll unmute you. So you have a question that's come in asking, does decay class relate to larvae availability and or extraction difficulty as predicted?
Well, let me go back to that slide. I mean, you would expect that, you know, in these later stage classes that it would be easier to extract. But I guess there's just probably less of those in the green forest. And that's kind of part of the reason why folks have such a high association of black-backed woodpeckers with burned forest, because they really, you know, they like these bark beetles that come in huge influxes after the fire occurs. And so that's why some of these results are so surprising, at least to me, that they're doing so well in the green forest, despite, you know, we maybe would not expect there to be the same amount of food resources available, and maybe the trees are also not as easy to excavate for them. I hope that answered your question.
Yeah, and we have another great question from Dave Shaw asking, do you think that heart rot in trees is important in both live and dead trees? Do you have any thoughts about that?
Oh, I'm sure it is, because if you think about where their cavity is, right? If you've got like a tree, they excavate, I don't know, a few inches in and then down into the tree. And so if there's some heart rot in the tree and they have some way, like a weak zone that they're able to get through the outer cambium into that heart rot, then I'm sure that would be an excellent spot for them to build their cavity. Thanks, Dave.
Okay, and then we can quickly answer the first question from Todd, and then I want to move on. But what cameras are you using for nest monitoring?
Oh, we've just got some little, they're just like little handheld camcorders that we put up on a tripod for one entire, basically like many hours on one day. And so they're all on like the 10th day of the, of after hatch, so that it's standardized to how much the parents are probably trying to feed those nestlings and that they're all the same age.
That's a great question. And then Todd, if you or others have questions, feel free to reach out to Mark. His contact information is in the program, but I want to transition us just so that we stay on schedule with our next presenter. So Jason, if you kind of want to get ready by sharing your screen and working through that, you should have those capabilities.
Thank you.
Thank you so much for kicking us off. It's kind of a put you on the spot there, the first presentation, but that was really fantastic. I'm just going to give a minute as folks potentially kind of shuffle between rooms, but things are looking good there, Jason. We can see your whole presentation, which is great.
Can you hear me?
And we can hear you too. So thank you. And Lucy, thanks for spotlighting him. A question from Madison saying, how do we shuffle? You're going to just have to leave the room and then go back to that webpage and join the other room. So a little less fun than getting coffee and treats in an in-person conference, but it does the trick. We've done our best to keep the presenters well fed and enjoyed over this day, but we unfortunately couldn't email or mail coffee and treats to everyone who's here, but we appreciate you guys being here. All right, so we are at 10:30, so I want to introduce our next presenter, it's Jason Piasecki. Jason is a student in the firm department, and he is going to speak today about red tree voles in working forests, particularly exploring what are the influences of occupancy for trees. So Jason, take it away.
All right, thanks, Sky. All right, yeah, as Sky said, my name is Jason Piasecki. I'm a master's candidate in John Bailey's lab in the firm department. And before I begin, I just want to acknowledge co-authors John Bailey and Katie Moriarty, and that major funding for this research comes from the National Council for Air and Stream Improvement and the Fish and Wildlife Habitat and Managed Forest Research Program. All right, and so I'm excited today to speak about our ongoing research on a little known and rarely seen arboreal mammal, the red tree vole. And there we go. Oh, still not responding. There we go. Okay, so red tree voles are endemic to Western Oregon, including the west slope of the Cascades and northwestern California. They're highly canopy-dependent species and subsist on a diet composed of conifer needles and fresh twig cambium. Now, because they spend a majority of their time, their whole life in the canopy and are usually only active nocturnally for about an hour a day, they're extremely hard to study. And while a handful of research has been and is still being conducted on red tree voles, population parameters in managed forests and broad-scale ecology are not very well known. Tree voles promise to get a little political here following the 2019 U.S. Fish and Wildlife Service decision to not list the northern distinct population segment, where the agency cited that there is more available habitat across the distinct population segment than originally modeled. The Oregon Forest and Industries Council commented on the need for further research to address data gaps. And just as background information, the distinct population segment that was up for listing ranges basically from around Corvallis, just north of Corvallis, up to the Columbia. Our research will not only lead to a better understanding of the species, but help inform land managers on the function of working forests on red tree vole life history. And so tree voles are commonly regarded as an old forest obligate species. They're most frequently found and studied in stands over 80 years old. Multiple studies such as Meiselman and Doyle 1996, Swingle and Forsman 2009, and Forsman 2016 also found that red tree voles do occur in younger managed stands, which is kind of the premise for this whole project here. And just for reference, the stand on the left there is 30 years old, the center stand is about 65, and the right stand age is about 98. However, we survey anything up to about 320. Our research does focus primarily on managed forests, but we still include old forests for comparison. Thompson and Diller did publish similar research in scope and methodology in 2002. However, their focus was on abundance and nest site characteristics of Sonoma tree voles in northwestern California. So I'd like to just provide a quick overview of our objectives. Most of these objectives do focus on gaining a better understanding of how red tree voles are using managed forests. All of these objectives aim to stratify our results across stand age class and distance from the nearest old forest patch. For this old forest patch, we estimate to be over 80 years old and over 20 hectares in size. And we operate under the assumption that these old forest patches are occupied by red tree voles already. Our study range encompasses over 2,000 square kilometers of the Oregon Coast Range, which makes this one of the most broad and ambitious red tree vole research efforts to date. And then within this study range, we wanted to ensure an even spatial selection of stands, so we divided the entire study area into 4 regions. We work in collaboration with actually 8 different landowners, federal, state, and private, to select our stands based on certain criteria. And we made our final random selection from stands that were between 10 and 30 hectares, within 500 meters from a road, less than 5 kilometers from our LiDAR generated old forest layer, and unlikely to be harvested in the next 5 years. And so this gave us a pool of over 2,700 stands, which we then stratified by age class and distance from old forest. And it is worth noting here that there were a few age classes in our south central zone that just were not available to fit our criteria. So we do have a little bit of a gap there. All right, so that comes down to basically 6 major objectives concerning an elusive 25-gram rodent stratified across 2 major stand parameters over a study range that's about the size of the state of Connecticut. So we do have our work cut out for us. I'm going to detail our methodologies as quickly but as clearly as possible. And this begins with our methods basically for our stands under 60 years old. As we'll see in a few slides, detectability changes drastically as we move into old forest. And so each stand has randomly generated plots at 1 plot per hectare. So that'll be 10% of the stand. These plots are surveyed in their entirety, and this is where we can confidently observe changes over time. And we'll also survey opportunistically for nests between plots. However, we can't basically can't draw any time series conclusions unless we're finding things in plots. And so here we see that ground surveying is complete. Survey tracks are in red, and 3 plots have been randomly designated to have trees climbed regardless of nest presence. This helps us estimate our detectability from the ground and collect unbiased data on tree metrics for each stand. And so once the ground surveying is complete, we will climb all the nests that occur within the live crown, as well as our random climb trees. We collect a wide range of data, including nest metrics, evidence of tree vole activity, nest use by other arboreal mammals, and measurements on canopy connectivity. And then for nests where evidence of recent tree vole activity is found, we will install remote cameras to passively monitor any nest events which take place, and that just keeps going until the following season. Starting this year, we are also installing cameras on a random subset of nests, regardless of tree vole activity, and this helps us to increase our chances of capturing and quantifying any tree vole colonization events. Now, as I said, that was for stands 60 years and younger. Our major challenge is that pretty much all tree vole research inevitably runs into is the issue of detectability. We see a massive difference in stand structure and composition as stand age increases above 80 years. And this makes it even more difficult to survey for voles. And detectability from the ground in stands over 80 years old can be as low as 3%. And just for a visual there, stand age on the left is 33, and that's one of our 320-year-old stands. Trees are pretty big, can't see a lot from the ground. We attempt to optimize detectability in old stands by conducting random climb surveys instead of ground surveys. And so this method, while more time and energy intensive, has the potential to increase the detectability of nests within the climb tree as well as surrounding plot trees. And so we can see here as an example, this nest shown on the left. is at a height of about 40 meters off the ground. It's barely visible from the adjacent plot center tree, which we were climbing, and could pretty much never have been seen from the ground. Now, when nests are found and climbed, they get assigned a tree vole occupancy status based on the presence of resin ducts, fresh Douglas fir cuttings, debarked twigs, and fecal pellets. And while tree voles aren't the only arboreal mammal to harvest branch cuttings for their nest, they are the only animal that eats Douglas fir needles. And so we can look for these resin ducts pictured here on the right to confirm tree vole activity at a nest. And a couple of you may be asking, what is a resin duct? The best analogy that I can come up with for everyone is just to imagine that tree voles eat only bananas and resin ducts are like banana peels. So in this analogy, the tree vole chews the banana peel off, discards it, and eats the middle. And so here we can see on the right, a fairly obvious tree vole nest and associated resin ducts. However, signs of vole activity are not always obvious even while climbing. This nest was likely constructed by a Humboldt's flying squirrel. However, meticulous inspection reveals fresh resin ducts inside the nest chamber, meaning that it was recolonized by a red tree vole at some point. As you may begin to gather from previous slides, tree vole nests come in all shapes and sizes. From an observational standpoint, nests found in old forest are generally more cryptic, including moss mat and cavity nests, and are difficult to detect even while climbing. And nests found in younger forests commonly occur on branch whorls and available tree deformities such as broken tops and split trunks. All right, moving into some quick numbers now. This project has been running for 2 field seasons, 2019 being our pilot and 2020 being a full 6-month season. Despite complications that came along with 2020, we safely and successfully completed 40 stands, surveyed over 3,400 trees, climbed 346 nests, took a lot of nest photos. And before we get into the next few slides, I would just like to caveat that the following plots simply represent preliminary results. We're not too deep into data analysis yet. Here, I just wanted to highlight changes in observed nest density. This is nests of all species across stand age and canopy visibility across stand age on the right. On the left, we can see that nest density decreases as stand age increases, despite the fact that visibility from the ground into the canopy increases along that same gradient. So this is interesting because it has a potential to add evidence to the presumption that tree vole occupancy may decrease as the canopy begins to open up in the 50 to 60 year age class. Here we just combined a scatter plot and box plots to show how the proportion of nests with evidence of tree vole activity changes across stand age. And this is kind of in following with the previous slide, we see a decrease surrounding the 50-year age class as that canopy opens up. This may be a little confusing at first, but I just wanted to focus on how nesting platforms change across stand age class. We are looking into available structures. Platform types to focus on here are broken tops shown in red, which we found to be most utilized in the younger age classes, and cavities and epicormic branch whorls in blue and purple, which we found most utilized in older stands. Now, it is worth noting that both cavities and epicormic branching do not become readily available in Douglas fir forests until trees mature beyond 60 years. And lastly, I wanted to show differences in estimated nest surface area across age class. This violin plot, the widest area represents the highest probability of occurrence for estimated surface area for each age class. And so we can see that generally we have larger nests in younger forests. Perhaps slightly more interesting than graphs of data are observations of changes in nest status over time. While nest predation and extirpation, such as the one depicted here, are normal occurrences in an ecological sense, they're difficult to observe and quantify on a temporal scale. And harder still to observe and quantify are nest colonizations. And this is honestly, most often it's just a matter of luck. In this case, we climbed a Humboldt flying squirrel nest in year 1 and found that the nest had been colonized by a tree vole in year 2. However, these types of events are pretty much invaluable in understanding how tree voles are using managed forests. And here, just wanted to include some other bonus participants in the study. Steller's jays and Humboldt flying squirrels were by far the most frequent nest visitors. Douglas squirrels, Townsend's chipmunks, and what we suspect to be a northern saw-whet owl showing up only a few times. Interestingly enough, we did not detect any weasels at nests, which have been observed in association with predation at red tree vole nests in Linnell et al.'s 2020 research. Okay, so in summary, we're making a lot of progress on a very big study on red tree vole occupancy in managed forests. We're utilizing 2 survey methods, one being completely novel to optimize detectability of red tree vole nests. We observed low overall nest density and tree vole occupancy surrounding the 50 and 60-year age classes. And we also were fortunate to observe both colonization and extirpation events at the nest level. We do have a couple limitations. We have yet to actually test the efficacy of our old forest survey method. And we do need to address some data gaps in our sample size and study range. We are tackling some of these issues moving forward. We're filling out categorical and spatial gaps in our stand distribution. We're increasing, filling in survey gaps in the stand based on the previous year's track logs. And we're also increasing the sample of nests that we install remote cameras on to try and get at that colonization and extirpation. All right, I'd just like to thank all the members of our hardworking field crew, Eric Forsman, Jim Swingle, and Mark Linnell for their expertise on red tree voles, and Tim Laws for photo documenting us in the field, and then our contributing partners and landowners on the right. All right, I think there's maybe a minute for questions. Feel free to ask, or feel free to email me later as well.
Great. Yeah, let's try to see if we can squeak in one question for Jason if we have one. Oh, do red tree voles only eat Douglas fir?
No, we're surveying primarily in Douglas fir forests, so that's what we see. However, if you get into the coastal ecoregion where you see more hemlock and Sitka spruce, tree voles can and do feed on those types of needles as well. However, they're not interchangeable. So a red tree vole that eats hemlock will not go eat Douglas fir, or vice versa. Pretty interesting.
That is really interesting. And so Dana Warren has a whole suite of great questions, but I think for the sake of time, I am going to have you guys connect over email for you to kind of chat about those questions with him. And
yeah, sounds great.
To our next presenter, Rachel. So Rachel, you should be able to share your screen and kind of get that rolling.
Great, thank you so much, Sky. Can you guys all see my screen?
It looks great. I'm just going to give folks like 30 seconds or a minute to do room shuffling while we play through musical chairs. All right, I'm just going to get us going. So our next presenter is Rachel Zitomer, who's in the Forest Ecosystems and Society Department here in the College of Forestry. and she will be discussing how forest age and floral resource availability drive patterns of native bee abundance and diversity in intensively managed forests in the Oregon Coast Range. So take it away, Rachel.
Thank you, Sky. Yeah, thanks everyone for joining me. I am going to be talking today about drivers of native bee community size and diversity in intensively managed forests in the Coast Range. And I'm going to start off. Oh, I'm also having trouble getting to the next slide. Hang on one moment. There we go. I'm going to start off just talking a little bit about native bees and why they're important and why I think this research is interesting. So native bees are one of the most important pollinator groups, both for commercial food production as well as for pollination of naturally occurring plants. And in the context of commercial food production, even though honeybees are, which are domesticated species, are the most common and I guess economically important species, native bees provide really important insurance against honeybee losses, as we've seen in the last decades when we've had colony collapse disorder disrupting some honeybee domestic processes. They contribute, native bees alone contribute about $3 billion per year of crop pollination services in the US. I misspelled that on my slide. And perhaps even more importantly, is their role in natural ecosystems. So in a commercial ecosystem, bees and insect pollinators facilitate reproduction in about 35% of species, but in natural, in terms of natural vascular plants, about 90% of wild plants are insect pollinated. And so they're really, really important for providing gene flow in plant communities and overall stability in natural ecosystems. And within temperate biomes like the Pacific Northwest, native bees are the most important group in terms of how ubiquitous they are and how effective they are at providing pollination services. Surprisingly little, however, is known about native bees in coniferous forests. The vast majority of research has been focused on agricultural settings, and very, very little research has been done in the context of forests managed for timber and wood fiber products. And really no research has been done in the temperate forests of the Pacific Northwest. In a study that was conducted in 2018 by my advisor and collaborators, they surveyed participants at a meeting of land managers and foresters to ask them about what are the perceived knowledge gaps in terms of pollinators in forests that are holding them back. And as you can see from this response graphic, many of them said that they are lacking basic information about the ecology of pollinators in forests, including what species are present, what their habitat associations are, and things like that, as well as how management affects these populations and communities. So my goal in this research is to try to address some of those knowledge gaps, to lay the ground for the ability to strategize conservation management decisions for this group. So the question I am asking in this particular analysis is fairly broad. It's really just how do the characteristics of regenerating forests influence native bee communities? And the factors that I examined were the age of stands, which is time since harvest and replanting, floral resource availability, which is important because, as you all probably know, that is what bees eat. They feed on pollen and nectar from flowering plants, and the landscape context in which bees occur in forests. So my first hypothesis is actually, I guess, two hypotheses, but I put it together into one for the purposes of of presenting, is that stand age and foraging resources are key factors in driving habitat quality for bees in forests. Bees in previous studies have been definitely associated with more open habitats with greater light availability and warmer ambient temperatures, which makes sense because bees, for one thing, navigate using polarized light, and they are, they are not mammals, so they require warm temperatures in order to go out and forage and be active. And also light availability is related to openness, and that promotes the growth of the floral resources that they feed on. So using that background, I hypothesized that stand age would be a big factor in driving the suitability of habitat for bees in managed forests, and that specifically as stands age and get older and canopy closure occurs, that bee abundance and species richness would begin to drop off. And for reference, in the left-hand side, I believe this is a 3-year-old stand, and the one on the right, I believe, is somewhere around 22. So you can see that there's a big difference in the overall structure of these habitats, of course, in the amount of flower resources we can see even from this distance. You can see this sort of blanket of yellow flower resources on the left, and not much happening on the right in terms of flowering plants in the understory. And secondly, I hypothesized that even once forest age was accounted for, that flower density and species richness would also be positively associated with bee abundance and species richness. My second hypothesis is about habitat amount. This is what I was talking about landscape context affecting bee communities. So if you compare these two sites that I'm showing here with these little red dots, you can see that there's a big difference in the context of the surrounding landscape. So the one on the left, there's a lot of young forest surrounding that site in the center. And the one on the right, there's barely any. So in this circumstance, I would expect that native bee abundance and richness would be greater where the amount of habitat that is accessible to them from that site would be, so I'm sorry, I lost my way in that sentence a little bit. I hypothesized that abundance and richness basically would be greater when the amount of habitat accessible was greater. The things that I measured to assess habitat amount were patch size of the stand, as well as amount of early cereal forest in the surrounding landscape. I used a couple of different radii because there's different habitat uses in terms of dispersal and foraging range of different bees. So I tested a 500 meter, a 1,000 meter, and a 2,000 meter radius in this circumstance. And then I expected that as these stands get older and the habitat quality declines at the stand level, that context would begin to matter less and less. And so I didn't expect there to be a big effect of habitat amount in those poorer quality sites. Basically, once there's nothing provided at the local level, I just didn't expect the context to matter very much. And third, I have a hypothesis about how I think that bee communities composition will change. Basically, that I think that beta diversity among stands will be characterized by more nestedness as stands age rather than turnover. So what that means is that I expected to see a small subset of native bees in older stands, a subset of what was there when the stand was younger, rather than seeing a different set of species that arises in older stands versus younger stands. So to test these hypotheses, we selected 60 stands that were intensively managed Douglas fir dominated stands in the Oregon Coast Range across an age gradient that is roughly representative of the rotational age in these forests that were selected from lands owned by 4 different landowners, which use the sort of standard practices that are generally seen in industrial forestry in this region. So that includes typical herbicide, nutrient addition, genetically improved tree stock, things like that. And in 2018 and 2019, we did some sampling, and all of these stands sampled bees using 3 different methods. We used blue vein traps, hand traps, and hand netting. Each of these sampling methods has some bias in what is collected, so we combined them all to get a more comprehensive view of the community. And we conducted flowering plant surveys, measuring density of blooms, as well as diversity of plant species to assess resource availability, and also measured canopy cover in every stand. So in terms of my analysis, I used generalized linear models to measure the relationships of native bee richness and abundance, which are my response variables, to the habitat characteristics I mentioned before, stand age, flower diversity and abundance, and habitat amount, which again is measured by stand size and amount of early cereal forest in the local landscape. And then to get at that second question about how communities change as stands age, I used beta diversity partitioning to look at the degree to which dissimilarity among stands of different ages is driven by species turnover. In other words, a new species occurring in different age stands, versus nestedness, meaning that it's the same set of species, but there's just fewer of them in older stands. What we found after we did all of this collection is that we sampled over 12,000 native bees in these forests, which is quite a lot. These represented 156 species or morphospecies, all 5 major families that are present in the Pacific Northwest, and 24 genera of bees. You can see that there's a pretty big difference between the composition, or not just the composition, the abundance of what we collected in between the 2 years. In 2019, or in 2018 compared to 2019, we collected about 4.5 times more bees and 42% more species. Getting some little pop-up notifications. What I found when I analyzed these relationships, the relationships of the bees that we captured in those stands to habitat characteristics. Sorry, one moment, my computer's having a little meltdown here. Was that a 5-year increase in stand age was associated with an average with a decrease of about 60% in mean bee abundance, and a decrease of about 50% in mean bee richness. So stand age is definitely having a huge effect on the size and diversity of these communities. We also found that once stand age was accounted for, a doubling in flower density increased mean bee abundance by 20%, and was associated with a 16% increase in bee richness. Neither response variable really related very much to habitat amount. So it looks from this like the relationships that we're seeing are really driven by local factors at the stand level. And even though we saw that big difference in abundance of bees between years, these relationships with the habitat characteristics remained constant between them. And lastly, when I looked at how the composition of these communities changed with the age of stands, I see that overall beta diversity, this is pairwise pairwise comparisons between diversity in stands, increases as the age difference between stands increases, which is to be expected, but isn't terribly informative. What you can see when we partition this beta diversity relationship into nestedness and turnover components, however, is that turnover is pretty steady through across the entire stand age gradient, whereas nestedness increases markedly. So what that means is essentially that, as I predicted, the beta diversity trends across these forests is characterized by nestedness, meaning that as forests age, the species that we see are a subset, a nested subset of the species that were present in younger stands. So finally, just to wrap everything up, what we've learned is that managed coniferous forests are home to a surprising diversity of native bees. It's quite a large number of species, even compared with other natural habitats that are more typically studied in relation to native bees. That native bee abundance and species richness is driven in a major way by stand age, as well as flower resource availability, and doesn't really appear to be affected much by habitat amount. And lastly, that species are lost as stands age, as habitat suitability declines. Some species basically drop out rather than new species being associated with older stands. What this means in terms of management implications to my eye is that there's really quite a short window of habitat quality in these landscapes in intensively managed forests, and that probably a big way to I guess to improve outcomes and improve and increase and promote native bee communities would be to lengthen that window of time by kind of slowing that progression from harvest to canopy closure that is really accelerated in industrial forests. So I think that is all I have to say. And with that, I will say thank you to everyone who helped me with this project, especially including folks who helped collect data, identify bees, and let us use their land for 2 summers, as well as our funders. And I will be available for questions.
Thank you, Rachel. Let's see if we can take maybe one question for Rachel, and then if you have others, feel free to email her. Again, that emails are in the program on that website. Do we have any questions for Rachel? Rachel, do you feel like you're starting to lose the rare species at a different rate than you lose those generalist species as canopy closure is happening?
That's a good question. I haven't really looked too much at the functional traits of these species yet. That's something I'm planning to do in the future. But my sense is that most of the species we see across all of these stands are fairly generalist, but that the less common species are the ones that are lost first, if that makes sense.
Cool. All right, so next up is Dustin Gannon. And so Dusty, if you want to kind of transition into sharing your screen and getting your presentation up in this last kind of minute as folks shuffle rooms.
Okay.
This is a great set of presentations, and so far I haven't heard lots of complaints that things aren't working, so I'm going to assume that the website is working all right for people, which is exciting.
Sky, can you see my mouse if I move it around?
I can't see your mouse, so yeah, I think,
Awesome.
yeah, I can see your pointer and your screen looks great. So we'll just move right into this, but our next speaker is Dustin Gannon, and he's actually joining us from the Botany and Plant Pathology Department. So glad to see some college and departmental diversity here. And he'll be talking to us all about his work pairing automated mark recapture and social network models to explore the effects of forest encroachment on hummingbird foraging patterns. So, all right, Dusty.
Yeah, thanks, Sky. Thanks to everyone for being here, and also to the organizing committee. I know it's a lot of work getting this all going. But yeah, I'm excited to share some of this stuff. This is some things I've been doing with doctors Sarah Fry and Adam Hadley, who are both Betts Lab alums, so from the Forestry department. And they initiated this, this research in the H.J. Andrews experimental forest. So I'm going to talk a little bit about that. Here in the Cascade Mountains, we have a bunch of high elevation alpine meadows, and they host a pretty wide diversity of flowering plants, as well as lots of pollinators. But recently they're being invaded or encroached upon by the forest. And I don't think we really know how these were maintained in the past, just that they are starting to close in. And what happened, one interesting thing that kind of happens as the forest encroaches is it kind of cuts off and isolates some portions of these meadows that used to be connected as one large meadow. And we became curious as to whether the plants that probably still exist in these small openings were still receiving pollen flow from kind of the main meadow. So a fragmentation sort of question, but rather than human driven, it's kind of could be climate or lack of fire or something like that. But the main question is whether pollinators are moving across these meadows now that we have sub meadows coming off of this main meadow, or whether they just kind of stick to the largest open area. And we came up with a couple ideas on how pollinators might respond to this forest encroachment. And we're going to focus on hummingbirds, and I'll explain why in just a minute. But the first idea is that the forest could kind of act as a barrier. If pollinators are flying low and visiting flowers kind of sequentially, and once they get up to the edge of the meadow, they just kind of turn around and head back towards the open area. So this would be a barrier. We call this a barrier hypothesis, and that this would reduce visitation to both plants that are at the front of the woody invasion and get overgrown by taller woody plants. And it would kind of reduce pollen flow across meadows that are newly isolated from the rest. Alternatively, I mean, hummingbirds are quite good flyers, as we know. So they could simply just fly over the canopy to get to other openings and other meadows that have resources. But this might reduce resource discovery and detection at the edge of that woody invasion, those plants that are newly overgrown and kind of hidden when the bird is flying high. So in this case, hummingbirds may act to maintain connectivity, functional connectivity of this landscape. However, we would expect a reduction in the number of breeding individuals in the population, because these plants are no longer being visited and contributing pollen to the population. But how do we measure foraging patterns? Pollinators in general are quite small, so we can't really ask them to lug around tracking units. Most tracking units are too large. So Adam and Sarah came up with this pretty cool idea, I think it's pretty cool, of using PID tags with hummingbirds. Hummingbirds are just large enough that we could use this automated mark recapture and insert a passive integrated transponder underneath the skin. And so here's a picture of Sarah actually doing that with this is actually a rufous-tailed hummingbird from Costa Rica. They did this in Costa Rica as well. But we did rufous hummingbirds in the H.J. Andrews, and then they set up these recording stations. So basically, there's a copper coil that's fixed around the entrance for or where the hummingbird would access the nectar in this hummingbird feeder. And when the hummingbird lands and puts its head down to drink the sugar water, then the pit tag passes through this copper coil, and it sends a signal up to this receiving box, which holds a data logger like a mini computer, and it records the timestamp as well as a unique ID for the individual that's there. And then we set up these arrays. So we had these arrays of 5 feeders, and we had 4 of them placed on 4 different mountains, so different summits within the H.J. Andrews. And we placed them so that we could have one large feeder in a central open area, and then we could have satellite feeders, that kind of represent different contexts. So one would be in another open area, but separated by some forest. We could have at least one that's inside the forest, but not separated by as much forest in between. And then also at least one that's completely connected to this center feeder. And then we can have kind of different contexts connecting all of these together. And our objectives are basically to estimate an effective distance on movement, basically to get an idea of how far hummingbirds move on average and how far pollen is likely to be dispersed. And then secondly, test for an effective local cover on the movement. So basically, what's the effect of putting the hummingbird feeder inside the forest on movement to and from that feeder? And then test for effective intervening forest as well. So this would kind of support that barrier hypothesis if we have a negative effect of having lots of forest in between 2 feeders, regardless of the local conditions. So we recorded a bunch of relocations of 59 different individuals. We have 20,000 relocations approximately. There were about 2,000 movements made by 33 birds. So a movement just constituted a bird being identified at a feeder and then another one within the same day. And we envision these data or the system essentially as a graph or a network, as they're often called. And we assume that this network was weighted. So what I mean by that is the thickness of these lines actually reflects the number of movements between them, and that it was directed. So, we have directionality here. The rate of movement from 1 to 2 isn't necessarily the same as the rate of movement from 2 to 1. And then we basically just put a logistic, or not a logistic, generalized regression model on those rates. But I'm gonna just highlight a couple things that I think are fairly interesting about these social network models that can, I think, actually be useful to elucidate some interesting biological patterns and behaviors. So as you can imagine, there are a bunch of dependencies in these data. So we have movements that originate at feeder 1 here and go to feeder 2, and those probably aren't independent of the ones that start at 1 and go to 3, because they both share an origin. Same thing is true for those that are coming from somewhere else but landing at feeder 1. So those are also not independent. And interestingly, so a standard way of kind of accounting for that is using what we call random effects for the feeder, essentially. But then we include multiple, one for the feeder as a sender and one for the feeder as a receiver. And those 2 random effects are also correlated, but they can be positively or negatively correlated. And what's interesting about that biologically is that, the direction of that correlation can actually kind of elucidate some source sink dynamics in the system. And that's not really relevant for our spatial or temporal scales in this project, but you could imagine this being useful in a larger scale project where you would have, a lot of individuals leaving a certain patch, but not so many returning. That would be kind of a source, and that would be negatively correlated with basically these. So the sender and receiver effects would be negatively correlated. But more relevant to our study here, we have the opportunity for basically correlation within a dyad, so basically 1 to 2 and 2 to 1, these could be either positively or negatively correlated. If they're negatively correlated, we're working with hummingbirds, and some hummingbirds are known to or thought to trap line. So they have these kind of routine circuits that they travel in. And if this was a directional circuit through this system, we would actually expect a negative correlation within a dyad. So basically these models, by that feature that we can have negative correlation within these random effects. And I'm not going to talk too much about these details, but we basically just gather these random effects up in a random vector, and we assume that they're not independent, but rather they come from a joint distribution with a bivariate normal with the opportunity for correlation. And that can elucidate some interesting biological features. But what we found is high positive correlation within a dyad, indicating lots of out and back movements. And we think that that could help maintain bidirectional pollen flow within these systems, as well as maintain functional connectivity. And so this is kind of a network map that the thickness of the line actually reflects the number of movements that we did see. So you can see a lot of movement within a given array, but not so much across these different meadow complexes. All right, and I'm going to go through a couple features or a couple predictions that we use the model to make these predictions. And this cartoon on the right of each of these panels shows the different scenarios that we're playing out here. So in each case, I'm going to show a light colored line, a light green line that just kind of shows our most open, most connected scenario. And then I'm going to show what happens if we kind of change the landscape in a certain way. And in this case, I'm assuming that these two locations, our two focal points, are still in the open. So these two plants or feeders are still in the open, but they're now separated by closed canopy forest. And then I'm asking the model to predict the probability of at least one movement per bird per week. As a function of distance, so we'll see that probability about goes to 0 as we hit 2 kilometers apart, regardless of the intervening landscape. But what happens is about a 54% decrease expected when we change from this scenario to the one on the right here. And similarly, we expect about a 65% decrease if we play out this scenario where these two locations are connected by open habitat, but they become overgrown. They're on the edge of the woody invasion. And then if we compound those two features, so basically lots of forest in between them, then we expect basically 84% decrease in the probability of movement between those two places. And so the main takeaway here is that we had a decent amount of uncertainty in these predictions due to relatively few feeder arrays. However, distance had a large role in all of this, and if forest encroachment kind of continues and increases the gaps between those open areas, then we don't expect very much movement between them, and that while our uncertainty is high, the magnitude or the effect size of these forest features is considerable, and that at the initial stages we have maintained connectivity by that high reciprocity in the movements, but again, as gaps increase, we don't expect much movement among those meadows anymore. Okay, and so there are a lot of people to thank because they were just pretty intense, I think, field work going into capturing those birds and maintaining those feeders. So a lot of people helped with field work. And then funders, obviously, and thanks to the BETS lab for acting as a sounding board on some of this stuff. And if you want more info on any of this, I have it all publicly available on a GitHub repository now, so feel free to check that out. And with that, I'll take any questions.
Yeah, great work, Dusty. So we have a question in the chat that said, did the weight burden of the pit tag have any noticeable effects on hummingbird flight or movement patterns?
It did not. Yeah, nothing noticeable. Yeah, good question. And I guess to add to that, this we verified, or I should say Adam and Sarah really verified that with radio telemetry in Costa Rica. So they were actually able to follow the birds and you know, say there's not much of a difference in their behavior.
We have other questions in the chat. If not, I know you didn't measure this in this experiment, but would you expect to see similar patterns with other pollinators in the system, like insect pollinators, or do you think that they might be responding differently to that kind of habitat fragmentation?
Yeah, I am nowhere near being an expert on insect behavior, but it is something I'd be interested in knowing. So I have another thing going where we have a bunch of genetic data on hummingbird pollinated plants in this area, and we also did a little exclusion experiment where we excluded those hummingbirds from the plants, but we weren't able to measure pollen flow across meadows in that case. We did notice there was basically no change in the pollen dispersal kernel when we excluded hummingbirds, at least within a meadow. Basically meaning insects are doing a lot of pollination within a meadow. What I don't know yet is how much they move among meadows.
Cool. We have a question from Lucy here saying, did hummingbirds that were already at the feeder affect patterns of other hummingbirds coming to the same feeder?
Yeah, I don't know that well because we weren't actually out there. The whole idea was this would be kind of a passive recording, so we wouldn't have to spend a bunch of time out there. But yeah, I mean, these hummingbirds are definitely territorial, but also persistent. So even though one might be defending that territory, you know, hummingbirds tend to sneak in and visit anyway. So I think there is some effect probably due to territoriality, but I think that's why we don't see a lot of movement beyond 2 kilometers.
Cool. Did you see any evidence in your data of the hummingbirds kind of trap lining your feeders?
So the high reciprocity, that high positive correlation of movement within a dyad, kind of indicates not so much. If we saw negative correlation, that would be more indicative of directional movement. But instead, it kind of went one way and went back. So there's a lot of kind of out and back stuff going on. So I guess from this, we can't say so definitively, but that's evidence that it's less trap lining and more kind of foraging out and back within a territory.
Great. Any final questions for Dusty at this point? All right. If not, we can kind of transition to our final speaker for this session, Jordan. So Jordan, if you want to share your screen and kind of get your presentation ready while folks transition, that'd be great.
Yeah, sure thing. My computer's a little bit slow, so it's probably a good idea to have it already ready.
Yeah, no worries. Dealing with technological issues is just kind of the name of the game with these sort of symposia.
That was so fast. Can everybody see that?
Yeah, it looks great. So
Awesome.
yeah, I think we should give it a minute or so to wait just so people can do their shuffling. I don't know if they are. I think people are.
No worries. Just let me know. I can't see my clock anymore. All right, there it is. Never mind.
Right. I'm in a conference room at OSU in the new PV, and there's a huge clock in the room that's off by 6 minutes. So it makes moderating just a little funky. But I guess it's just when the conference rooms don't get used, no one looks at the fact that the clocks are very wrong.
Did they manage to change it for daylight savings?
They did manage to change it for daylight savings. They just missed 6 minutes somehow. So who knows? It's just goofiness. All right, well, it's now 11:30 according to my correct clock, so I think we should go with that. So this is Jordan Ellison is our final presenter for the day. And before I introduce her, I want to remind everybody that after this, there'll be a poster session that starts at noon, so you get a tiny bit of a break. But I encourage you to go onto the website, can you peruse those posters, pop into people's Zoom rooms if you have a chance to do that. They're open for half an hour in 2 slots. And if you don't feel like you can kind of hit that, you could check out the posters this afternoon or later and still send people feedback, either using those feedback forms or just email. And then this afternoon, starting at 1:10, we have another set of concurrent presentations. So lots of great graduate research to enjoy today. But our final presenter for this session is Jordan Ellison, and she is a student in the firm department, and she's presenting on the conservation value of slash piles for Pacific martens and Pacific fishers. So take it away, Jordan.
Awesome, thank you so much. Hi, everyone. Again, my name is Jordan, and I'm a master's student in forest engineering and resource management. And I'm here to talk about the research that I'm working on with my advisors, Dr. John Bailey and Dr. Katie Moriarty, investigating the conservation value of slash piles for Pacific martens and fishers. I'd also like to recognize Dr. Angela Larson-Gray with INCASI, who's provided us with a ton of guidance and has been a huge help to me on the small mammal portion of this project. I'm going to start by introducing our focal species, Pacific marten and Pacific fisher. For those that are not familiar with these guys, they're small forest carnivores in the family Mustelidae, and they have fairly broad ranges in the forests of North America. But here on the West Coast, populations have declined over the last century and appear to be somewhat isolated. And that's resulted in some populations having received recent federal and California state protections. And there's basically 2 key pieces of information about martens and fishers that suggest that slash piles could be used as a tool for their conservation. The first is that we generally expect martens and fishers to use clear cuts less than areas that are forested. And both species really show some hesitancy in venturing out into openings. And this might be because the increased exposure elevates their risk of predation. And ultimately, this likely limits their ability to move across the landscape and could create barriers to connectivity. The second point is that large trees and snags and woody debris are really important structures for martens and fishers. Individuals will use multiple structures as they move throughout their home range, and they provide thermal insulation, escape cover, and protection from predation generally. And they're really especially important for reproductive females during parturition and kit rearing. And the added structural complexity from coarse woody debris may also contribute to hunting opportunities. So these points are important here because, as you might imagine, they make martens and fishers somewhat sensitive to forest management activities that create openings and reduce the recruitment of large woody structures, as well as large-scale disturbances such as stand-replacing fires. Martens and fishers have both been observed incidentally at piles, and on the Klamath Plateau in Southwest Oregon, GPS-collared fishers used slash piles for resting and as maternal den structures. It's not really well understood how frequently martens and fishers use slash piles or what benefits they might provide, but it seems reasonable to think that they might provide a similar function to naturally occurring woody debris. So our goals then are to begin to quantify martens and fishers' use of piles. At this stage, this basically looks like testing their willingness to visit slash piles in clear cuts and how that relates to stand and pile characteristics such as stand age, vegetation, distance of the pile from important landscape features, and the size and composition of the pile itself. And in addition to direct marten and fisher use, as I mentioned, coarse woody debris may increase hunting opportunities. And that's because woody debris is also associated with increased small mammal diversity and abundance. So in the marten fisher context, basically what that means is that they would have a larger and more stable prey community, since small mammals typically make up the large majority of marten and fisher prey items. But we also hope that our findings from this project might inspire additional research as to how slash piles might benefit other species and taxa. And last, as we all know, our forests are increasingly threatened by fires, and part of the reason for pile removal is to decrease the risk of wildfire spread. So in addition to documenting the potential benefits of retaining piles, we plan to simulate the risk of fire posed by retaining piles, with the goal of providing some guidance regarding the trade-off between conservation value and risk. So our strategy can be thought of kind of in 4 parts. First, we want to obviously document martens and fishers at piles. Second, we are performing small mammal trapping to assess whether piles support an increase in prey communities and thus increased hunting opportunities. Third, we are estimating pile and stand characteristics, including pile sizes, composition, distance from landscape features such as roads and streams. And finally, we'll be doing vegetation and woody debris sampling in stands with slash piles to assess the cover that's available and the risk of fire. So during our 2020 field season, we surveyed 35 stands, and our study area was entirely on land owned by Green Diamond Resource Company in Humboldt and Del Norte counties in Northern California. We chose this area for a couple of reasons. There are populations of both fishers and martens here, including 2 of the 4 populations of Humboldt marten recognized by the U.S. Fish and Wildlife. And due to a combination of air quality restrictions and the Humboldt Marten Safe Harbor Agreement, piles have been retained over time, creating this really great opportunity for us to survey piles of different ages and sizes. Our study includes 3 treatment types, and the main focus is on these first 2 which make up stand pairs of a regenerating stand with slash piles and an adjacent older forest. And we're defining older pretty loosely here, as you might see, since the key thing that we're interested in here is whether a marten or fisher will leave the cover of the forested stand to come to a slash pile in a clearing. The third treatment is a regenerating stand that is away from any slash piles or has had slash piles removed. And the purpose of this stand is to look at small mammal communities away from the influence of piles. So our camera surveys take place in just those first 2 stand types that I described. We typically survey 1 pile per stand, and each pile is surveyed using 3 evenly spaced cameras around the perimeter of the pile. The piles are not baited with any food reward, but we do use the olfactory lure Gusto within the frame of each camera. In addition to pile cameras, we set 2 baited cameras in the adjacent forest within 50 to 200 meters of the edge of the regenerating stand. And the purpose of these cameras is just to determine whether there were martens or fishers near the regenerating stand during the survey period. It's obviously not a foolproof method, but at least gives us some idea. We're also working with rogue detection teams to do scat detection surveys in addition to our camera surveys. And the really great thing about working with scat detection teams is that it's a passive detection method. So we can detect martens and fishers without influencing their movement with bait or lure, and they effectively survey all the piles in the stand rather than just one, which is just a couple of the key benefits over camera surveys. And to maximize our detection capabilities, most of the scat detection surveys overlapped with camera surveys, with another handful of stands surveyed exclusively by scat detection, just to increase our sample size. Additionally, we can use those collected scats to compare marten and fisher diets to the small mammal species that we're finding at the piles. So because small mammal trapping is a fairly intensive part of our study design relative to the other aspects, we only trap in a subset of our surveyed stands, and that subset is stratified by age, latitude, and pile size. Each trap session is 4 nights, preceded by a minimum of 6 nights of pre-baiting. We're using this novel web-shaped trap configuration. This has basically 2 main benefits in that we can achieve fairly robust density estimates, and this configuration that we use for slash piles is adjustable to accommodate piles of different shapes and sizes. Our vegetation sampling takes place at 3 randomized points in each stand, a total of 6 plots for a total of 6 plots per stand pair. At each point, we collect understory vegetation data within a 100 square meter circular plot, and overstory data is collected within an expanded plot of 500 square meters on that same center point. And then again, from the same center point, we collect data on woody debris using a modified Brown's transect of 20 meters per sampling plot for a total of 60 meters sampled per stand. Our woody debris estimates will go towards informing both our fire simulations and gaining insight as to the woody structures that are available to martens and fishers on this landscape at this time. I'm just going to talk really quickly about what we've seen thus far, and I want to emphasize that these are all preliminary results since we are in the middle of the study, but the big highlight from our first field season was that we did get several fisher detections at piles. Of the 35 stands that we surveyed, we detected fishers at 13 stands in total. We did not detect any martens at piles, but we only had 1 marten detection overall, so we really haven't learned anything about martens in this context at this point. And so far, we're not really picking up on any trends on some of the things that we thought might be making a difference in pile visitation, such as stand age or distance of the pile from the forest edge. We completed 8 trap sessions in which we captured 405 unique individuals from 12 species. And this plot basically just shows us how similar or different community assemblages are between regions and treatments. And as you can see, there's not really a clear pattern emerging quite yet as a result of the treatments, but we do see some regional differences. And we can see here that small mammal diversity tended to be relatively high at piles, but we had a lot of variation by region here again, so we can't really draw any conclusions about this at this point. Our most common captures by far were deer mice, followed by chipmunks. That's probably not a surprise to anyone who's done small mammal trapping. Generally speaking, the relative abundance of those species were higher in the regenerating stand than in the forest. But whereas we saw a drop off in deer mice abundance at the slash piles relative to the regen, the relative abundance of chipmunks was actually fairly comparable at piles and regen. So the main takeaways from this first field season are, for one, we did find that some fishers were willing to visit a pile in a clear cut when incentivized. While we didn't see any trends in the stand age or distance to the forest, we have not yet explored the relationships to vegetation, and it's possible that the fast-growing coastal vegetation plays a role by providing cover, which is not going to be consistent across their range. So that's something to consider when thinking about these results in the context of other populations. The next point is that the effect of piles on prey communities are not yet quite clear in our study. We know that a similar study in British Columbia saw that harvest units with large piles saw increasing abundance and diversity of small mammal communities over time, but we just don't have a large enough sample at this time to determine whether we will see a similar trend. It does seem likely that the effects differ between species, and that could be really important if we see that piles support important prey items like chipmunks and voles. And most of all, we know that we need more data. And thankfully, we have an upcoming field season starting in June. And we'll be working in California, and that part of the season will look really similar to the 2020 season. But with a bigger crew, we have some hopes to fill in some data gaps, particularly in stand ages, as we found during the 2020 season that the younger stands were quite a bit more accessible and ended up being overrepresented in our sample, whereas stands in the 6 to 15-year age range were frequently disqualified due to either time constraints or safety concerns. And we haven't really learned anything about martens here. And since it is our goal to collect data on both species, this coming season, we plan to spend some extra time in the areas known to be occupied by martens to try to fill in that information. We also plan to expand surveys into Southwest Oregon, and here we'll focus on vegetation and woody debris sampling in these areas that are occupied by martens and fishers. And we'll continue to work with rogue detection teams and have them doing some scat detection surveys there as well. And last, we will be returning to the Klamath Plateau and revisiting rest sites and collect measurements on those piles that were used by GPS collared fishers. And of course, I would like to acknowledge the many people that have made this possible and provided support to the project. All the folks at Green Diamond and Nkasi who provided enormous field support and other technical support, as well as the rogue detection teams. And I would like to give a special thanks to the Fish and Wildlife Habitat Managed Forests Research Program for making it possible to bring this project to OSU. And with that, I will take questions.
Thanks so much, Jordan. That was a wonderful presentation. So questions in the chat, or you can raise your hand and unmute yourself, or I can unmute you. But maybe while folks are getting those in, I had a question. If you've done the kind of scat detection yet, did you see visitations at piles that you didn't kind of bait with the scent?
We have done some scat detection work already. Like I said, we sort of contract that to rogue detection teams. So, and I haven't gotten that confirmed data from them yet. So I don't think that we did actually, but it'll be hard to know until we get the genetic confirmation.
We'll just have to stay tuned for 2021 or 2022.
I noticed from a lot of the photos that the slash piles had a really wide range of wood diameter and just overall size. So I imagine there's a big difference in the space available inside them and the temperature. I was wondering if there's a lot of work on that.
Well, so generally speaking, there's not a lot of work on slash piles previous to this in general. But we, as part of our measurements of piles, we do estimate the proportions of the different sizes of the pieces, if that sentence made sense. Did that make sense? So we're taking estimates of piece size in the composition.
Great.
Yes.
Unless there's, oh.
Hey, sorry, Sky. Have you taken apart any of the scat yet to see the chipmunk makeup versus, the mice versus shrews and all that kind of stuff? And is that why mice aren't around the piles?
Say that again. What was it? I feel like that was two questions. One, have we done the scat analysis yet?
Right.
So I am not personally doing that. Katie sent those samples they're being genetically analyzed, and obviously I don't have the expertise to do that. So it's not like we're drawing them and picking them apart. We're doing a genetic analysis.
I was picturing, looking for their little skulls and all.
Oh yeah. I've done that too. I when I was an undergrad, I volunteered in Dr. Levy's lab, picking apart bear scats.
Yeah, and I had just noticed the drop off in mice at the piles, and I was just wondering if that was a prey phenomenon, you know, or I guess it would be a predator phenomenon, or if, you know, so is that competition or a lack of a food resource around the piles for them?
So it's hard to know. In my discussions with Angie about deer mice, is that the sort of conventional wisdom about deer mice is where other species don't tend to do very well, deer mice sort of dominate. And then you see a little bit of a drop off as in deer mice as diversity increases. And so it could just be that we're seeing others, it is more of like a competition type mechanism.
Great answers to those questions and really a fantastic set of presentations altogether. It's really incredible to me to see the research that everyone's working on. So I'm going to let you guys all kind of go to lunch, encourage you to check out those posters, give folks feedback if you haven't already. And we will see you guys in the afternoon for afternoon sessions. But thanks for sticking around today.